I'm just going to start very briefly with giving you a little bit of an overview of what Sectra does. So if we go to the next slide. So Sectra is a company and really has two main areas. One is within imaging IT and the other being IT security, but diagnostics and imaging IT is by far our biggest part of the company. Apart from that, we have a couple of smaller areas within business innovation, as we call it. And among those, if you click forward, we have genomics IT, which is sort of the focus of today. So if we go forward to the next slide. So really, diagnostics is becoming more and more integrated as IT systems evolve. And the diagnostic journey really stretches all the way from population-based data down to the realm of the molecular. So Sectra really started its journey in radiology roughly 30 years ago, but have since grown into most imaging-based diagnostic areas. And we talk a lot about enterprise imaging management today. And I think roughly 10 years ago, we started thinking about digital pathology, and we're one of the pioneers within that field. It took a while to make a good product out of it. But anyways, I think approximately 2 years ago, we started working on this piece, which is the focus of today, the genomics. We went on a journey to go outside of imaging-based diagnostics and really into perhaps the final and deeper level of diagnostic information. And that's what we're going to talk about today. So it's important to understand, though, that clinical decisions and finding the optimal treatment for a patient really revolves around having all the data from multiple disciplines. I mean, we can zoom in and use molecular data, and it's usually really accurate to pinpoint how to treat a patient. But we also need to understand what we're not seeing, sort of how that fits into the big picture, and having it all together is what's needed to help the patient. And when we talk about multimodal data, having data from all different ologies and disciplines, traditionally we think about values within research and AI development, but diagnostic information often also holds a clinical value outside its respective silo. For instance, as feedback loops, improving upstream diagnostic decision, taking the decision, do we proceed with more in-depth diagnostics or more detailed diagnostics? And if we go forward, so molecular diagnostics or precision diagnostics is really a key component of what's in the term precision medicine. I like to think about precision medicine as sort of like two halves of a coin, where the one being precision diagnostics, which is really what enables precision therapy. And together we have the concept of precision medicine. And as this slide tries to illustrate, at least, precision medicine is growing. So we don't need to look at the specific here, but rather just the trends. And it actually really doesn't matter, at least from my research, to what metric you choose to look at. I mean, it all points upwards, so regardless of being number of articles within the sort of matching the term precision medicine, or number of FDA approved drugs, or just the current and predicted market size of precision medicine, it's all pointing upwards, right? So this is something that's interesting to us and really needed in diagnostics. And if we go forward, for us, going into something completely new is, of course, not something we do on our own. It's really hard without a partner, and that's sort of what we've done in all the areas we've extended into. So we were really seeking a customer that had extensive knowledge in this space, that were willing to work together to build a new IT system for molecular pathology, which is sort of where we're starting in the area of genomics. So luckily we found Penn, and after, obviously quite some extensive preparatory work, we started this project and sort of went into an active phase of the project in September 2022. But to give you guys a bit of background of what molecular pathology and diagnostics really is, and to talk you through this really cool project that we did, it's a great honor for me to now leave over to Dr. Priore. Dr. Priore is the lab director for clinical cancer genomics at Penn Medicine, and really a key member of this project, a key faculty member at Penn, and obviously been instrumental in the design of this project. So over to you, Dr. Priore.
Thanks so much, Fredrik. I really appreciate the introduction. And I'm very pleased to be with all of you today to sort of talk about my area of medicine. So as a physician, I focus on using molecular techniques and molecular biology principles to really turn what Fredrik was talking about in the introduction to actionable data that change the trajectory of care for patients with cancer. And so, my specific specialty in medicine is called molecular genetic pathology. It's a very, very broad field, and I just want to note that because what we're going to be talking to today is very specific in that field. We're going to talk about cancer genomics. And so I want to give an overview of what cancer genomics is and why it's relevant for clinical care, and give you some background about what I do and what is involved with running a clinical cancer genomics laboratory. And that really feeds into an understanding of why we needed to embark on this collaboration together to kind of harness all the information that comes out of this laboratory to impact clinical care for our cancer patients. So we'll go through that, and then we'll talk through the Penn Sectra collaboration, what motivated it, go through the timeline, how we developed the product, and how we prepared to launch it for live clinical care. And then we'll go through some high-level feature review of the product itself and review the benefits and the clinical impact that we're seeing for that product. And then turn to the future and talk about how we're thinking about genomic diagnostics and how we're going to integrate and move things and push things forward to continue to innovate. So I'm gonna take a big step back and do a very broad overview of cancer biology. This is a whole semester worth of material in 5 to 7 minutes. So apologies, it's not comprehensive, and hopefully it just gives you an idea of kind of what we do. And so, genomics, when we talk about the genome, our body is made up of cells. The cell, it has DNA in its nucleus, and that is a genome. And the genome is basically a set of instructions for creating all of the molecules, specifically proteins, that do a lot of the functional things that make our organs and our tissues work appropriately. So this schematic just shows you, a cell with the DNA in the nucleus, and then portions of the DNA are areas referred to as genes, and genes code for very specific proteins. So genes are the instructions for creating proteins. This is a slide that shows this kind of central dogma of molecular biology. So we have our genome, which is our DNA. Our DNA is transcribed into another molecule called RNA, and that RNA serves as a template for the specific instructions for building a protein. And so we're all born with a specific genomic sequence, and most of the time our proteins work, they're made, the instructions for making them are correct, and we have proteins that work the way they're supposed to be. But unfortunately, as we age or as we become exposed to different elements, our DNA can be altered. So we think about acquiring mutations in our DNA, and they can come in a variety of flavors. This sort of outlines different things that can happen to the DNA. The details aren't so important. What is important is just sort of understanding that you can change the underlying DNA in a cell, and that can lead to problems. And I'll talk about kind of how that's manifested. So some of the simplest changes you can think about is that you change one DNA residue to another, you delete pieces of your DNA, or you insert sequences into your DNA. and that causes problems. And specifically when it comes to cancer, there are proteins within the cell that their main job is basically to control whether a cell is dividing or growing. And when you pick up mutations in one of these key residues, and I don't know if you can see my pointer or not. You can, okay. So here, this is a kind of a normal signaling pathway. And so when all the proteins are working the way they're supposed to, this regulates the cell, it grows and divides exactly the way it's supposed to. But when you acquire a DNA mutation, and this example is in the BRAF gene here, all of a sudden that signals too much and the cell grows out of control and it's not regulated anymore. So this is a very simplified view of how DNA mutations can translate into a disease. And so that's basically what I just said. You have a normal cell, you acquire DNA mutations in important genes in your genome, and that is what can lead to cancer. And cancer cells, when they grow, form tumors, and that is kind of in a nutshell cancer biology for the purposes of this talk. But why is that important? As a physician, why is it important for the care of cancer patients? So I really like to use the example of lung cancer because this slide sort of shows our understanding and thinking of lung cancer and how it's evolved over time. So if you go back 40 years and you got diagnosed with lung cancer, it was simply based on where the tumor was, right? So they had a tumor in the lung, you had lung cancer, you received whatever treatment was available, you received surgery. And as we started to get more sophisticated and looked at cancer cells under the microscope and started staining them with special stains, we started to subdivide tumors into more specific categories. So we go from lung cancer to groupings of lung cancer based on how they look under the microscope. And so this second pie chart shows you kind of the big umbrella categories for lung cancer based on how they look. And then as we started to understand what the underlying DNA changes were that led to this cell becoming cancerous in the first place, we start to see that the categorization of these or the classification of these becomes much more diverse and much more complicated. And this pie chart at the end shows you all the different genes that can be mutated and lead to a normal lung cell turning into a cancer cell. And so that's great for our understanding of cancer, but why is it important clinically? It's important clinically because as we understood these specific underlying molecular changes, therapies were developed that could specifically target a tumor that carried a very specific change in the DNA. And so this gives you a schematic of one of the genes that is recurrently mutated in lung cancer. It's called EGFR, and shows you a summary of some of the major changes that we see in lung cancer patients. And so all of that is to say that cancer genomics, understanding the underlying DNA changes that are happening in a cancer cell, facilitate precision medicine, which is what Fredrik was talking about in the introduction. And what is precision medicine? Precision medicine is treating the right patient with the right drug at the right time. So if we have a cohort of patients with lung cancer, we do this genomic testing on them. We can divide them up into groups based on the molecular classification of their tumor, and we can give them very specific treatments that optimize their care, extend their quality of life, extend their progression-free survival. And so this has really changed the paradigm in terms of how we address cancer, how we treat cancer. And it has made huge advances in terms of people's ability to live and live with a high quality of life, even with very advanced stage cancers. So that's great. These findings can change the management of patients, can be really impactful for treating cancer patients, but how do we realize that the ability to translate mutations that are in the patient's tumor into actionable information that the clinicians can use? So at Penn Medicine, we have a laboratory that's specifically set up to do this type of testing. It's called the Center for Personalized Diagnostics. And I won't go into the details too much in terms of technically how this works, but our CPD laboratory uses next generation sequencing or massively parallel sequencing to identify these underlying DNA mutations. So basically, we take genomic DNA from the tumor, from the patient's tumor. We prepare it in a very specific way so that it can be sequenced on this sequencing instrument. And next generation sequencing allows us to sequence not one gene, but actually hundreds of genes at the same time. And it also allows us to sequence dozens of patients together. So we can actually do this in a very high throughput way and get a lot of molecular information about a tumor from one single test. And so that's the big takeaway. The technology is not as important, but if you're interested, there's great kind of YouTube videos that sort of walk you through how this actually works from a technical perspective. Okay, so to actually operationalize this, there's a lot of things to consider, right? So we think about clinical laboratory testing, and that includes this clinical cancer genomic testing in terms of the phases of testing. So there's the pre-analytical, everything that happens before I do the sequencing, which includes getting the tissue from the patient, biopsying the patient, transporting that sample, getting the genomic DNA out of the tumor and making sure it's high quality and can be appropriate for the sequencing. Then all the steps involved in the sequencing itself, and then going through all of the data that comes out of that sequencer, analyzing it, interpreting it, and then reporting it in a way that is easily digestible for the clinician, and they know exactly what they need to do to treat the patient. So what does this mean? And am I finally going to get back to Sectra and what we did? Yes. The sort of implications of a laboratory or a testing paradigm like this is that we create huge amounts of data, and we have to organize a huge amount of information to make this successful, right? We have to receive orders for testing. We have to accession the samples when they come into our lab. We have to track them through their journey through our lab. We have to track all of the laboratory steps that are involved in that next generation sequencing workflow that I showed you on the last slide. We need to receive and process data from the sequencing machine, analyze it, and then we need something to annotate and curate all of the mutations that we find from the patient's tumor. And then finally, we need to generate a report that's easily readable and transmit those results to the patient's chart. So this requires a huge amount of IT support to make this successful. And to put this in historical context, clinical genomics is not that old of a field relative to other specialties in medicine. And so when this started to hit the scene in terms of these types of laboratories opening up, there was nothing available. Nobody had any products available that could handle this data. And so laboratories like the CPD and others were really left on their own to develop their own laboratory information systems, or LISs, to manage all of these workflows. And they're incredibly complicated. We used a kind of generic database software platform called FileMaker Pro to build out a database that could meet our needs. And so that was a homegrown solution that we managed ourselves and established some amount of internal competency with, but we were rapidly outgrowing it. So there's several problems with it. It didn't integrate with the other LISs that we used, nor did it integrate with the electronic medical record. It was inefficient and error prone. It didn't have PDF reports. It didn't have any auditing capability, so people could change things and you wouldn't know that they changed it. And that's a problem for maintaining clinical data, obviously. And it was maintained by staff and faculty. So namely, in the last few years, it was me and another person in the laboratory. And not that I'm not a capable person, I would say, but it's certainly not the ideal situation you'd want for a laboratory information system, right? You don't want the people in the laboratory maintaining that type of software. And especially since it's a resource drain and it pulled people from other tasks that they could have been focusing their energy on. So this was really the motivation for trying to seek out some solution that could help facilitate clinical genomics testing and meet our informatics needs and help us grow into the future. And so we luckily got partnered with Sectra, and that's sort of how this project got started. This is just a diagram to show you what I talked about on the last slide. FileMaker Pro before we switched to Sectra, FileMaker Pro was really not integrated into any of our other information systems, and so it was really just very problematic. And I just wanted to show this image because it'll be in kind of stark contrast to the current integration that we have once we completed the project. So there was a lot to think about in terms of the functional scope of what we wanted to accomplish with this collaboration. So at a minimum, it needed to achieve all the things that we already had built into FileMaker Pro, right? I've already gone through all of these tasks on the previous slide, but we had to build a product that could just serve the main tasks that we have to do to run the laboratory. But what we were looking for is not just to make a one-to-one replacement, but really to advance the state of what we were doing and how we were reporting things in the lab. So we wanted it to integrate with Penn Chart, which is our electronic medical record. We wanted it to generate visually appealing PDF reports, provide discrete data, and I'll talk more about that at the end of the talk, have audit capabilities, have system guidance that help the user really do their job efficiently and remove or reduce the opportunity for introducing errors into the process. And then additional benefits, taking the responsibility of maintaining the software and developing the software off of our clinical team and partnering with people who actually do this for a living is a huge additional benefit. All right, so this is a slide I'm going to spend a little bit of time on. It's very difficult to convey how hard this project was in a PowerPoint slide. And so I really want to just say, you can look at the dates to see, this started in 2020 with early discussions. It ended in 2024, or it launched in 2024. I apologize. We're still working together. But this really was a complicated problem to address. It required a very thoughtful approach. And once the sort of agreement was put into place, the real active part of this started probably in 2022. And that involved the Sectra team coming to our lab, sitting down with us for several days and watching every piece of the laboratory workflow and understanding kind of what the problems we had, where information flowed into the lab, where information needed to flow out of the lab, and how are we going to build a product that met all of those needs. So, the discovery and the planning was a big portion of it because even though FileMaker Pro was somewhat homegrown, it had evolved over several years to be incredibly sophisticated in terms of the number of tasks and the amount of information it was holding. So that was a big portion of it. And then when we went into active development, this was Fredrik and his team kind of taking all of that data, going and building the software from scratch, coming back to us and saying, does this work? Does it not work? And iterating over that for over a year, basically, and getting to a point where we had a mature enough product that we could say, okay, now we can start to operationalize this product. And that's when we kind of got into the very sort of heavy lift of saying, okay, now we're going to move out of active development and start to deploy this. And what did that require? It required a huge amount of coordination with both of our teams to say, to start to test every possible configuration to make sure that there wouldn't be any disruptions to clinical care. And so that involved doing integrations in test environments before we launched it into production. I think we did hundreds of test cases and sort of every permutation combination of things that we could think about to make sure that results were flowing through, that that every step of the workflow was working the way we thought it would be. It also involved a huge amount of paperwork and regulatory work and training. So we had to train every user in our laboratory to switch from our old system to our new system. We had dozens of documents that had been written in terms of how the laboratory worked that all reference FileMaker Pro. And so it took months to update everything to then have instructions for how to do all these tasks in Sectra. Even sort of little things that we took for granted took a lot of time, like making sure that Sectra could communicate with our label printers so that we could label our specimens appropriately. And so this was kind of a very, very intensive, months-long operational preparation phase. But thankfully, it led to a very successful go live a few months ago, and we were very, very happy with that. And there was minimal disruption to our clinical workflow, and we very kind of smoothly transitioned from our old system to our new system. But I do want to highlight, kind of the magnitude of this this task. There was 6 to 10 people on the Penn side kind of working on this for a substantial amount of their time. Fredrik had a dedicated team of 6 people that was developing the product on his side. And it wasn't as simple as just starting and flipping the switch on the new system. We had to think about making all of the data that was in our old system available in the new system. And so we ended up having to migrate 7 million variants and over 40,000 clinical cases, historical cases that were in our old system to our new system. And so this was really just kind of a very monumental task. And considering everything that needed to be done, I see Fredrik shaking his head. I think we did it in an amazingly fast time frame. So once we developed the product, this is coming back to, what is our data integration now that we have the Sectra product? We can see that it's completely integrated into our EMR. So now orders flow from our EMR when they're placed directly into the Sectra system. And then all aspects of the laboratory phases of testing are run in the Sectra system. The Sectra system communicates with the software that processes all of our sequencing data. So it sends it out, and then it receives the results back. And then it has all the functionality we need to report, and it has an interface to then return the results back to the chart. So very, very nice integration that we just did not have before. So I just want to show several sort of screenshots that highlight the realized benefit of deploying this project. So this is just showing you what the queue looks like. So these are the orders that are coming in automatically from the EMR now. And previously we'd have to manually enter every single one that came into our lab. And so now this process is largely automated. And just so everyone knows, this is there's no protected health information on any of these. It's all contrived names, MRNs, things like that. So please don't worry that I'm divulging any patient information here. This is all test environment cases. And so you can see the system nicely organizes our cases, so we can see what the tumor tissue is, where it came from, We can see on the left-hand side the patient history. So we have a record of all of the historical and current testing that's occurring for the patient. And then on the right, we have an audit trail. So we have a very, very clear history of everything that happened to the record. So we know who changed any of the fields, when they changed it. And this was a level of auditing and accountability that we didn't have in the previous system. And this is really important to make sure that quality is maintained and consistency is maintained when you're dealing with patient information. This is just showing you the laboratory component of the software, and I won't show every single piece of it. It's pretty complicated, but the takeaway is that it automatically shuffles the samples that are ready for the next step of their laboratory journey into the appropriate place. And it makes it very easy for our laboratory technologists to say, okay, these are the samples I have that are ready for DNA extraction. I highlight them, I hit start, and it does a lot of the work for setting it up for you. And that's shown here. You can see it even gives very nice visual guidance. So this little grid with the circles on it actually is a visual representation of the plastic plate that the technologist has in front of them on the laboratory bench. And so the system gives guidance in terms of tracking which patient sample went into which well, and tells you what to add to which well and how much you should add. And so these cues and this integration step by step through the lab workflow makes it more efficient for the technologist, makes it less error prone, and helps with efficiency, which is wonderful. And it even gets broken down. We can customize each of our tests to sort of break it down by day. And so it tells you exactly what happens on day 1, day 2, what are the tasks, what are the specific components you need to have to do the testing, and how much you need to add to each sample. And so a lot of the manual work that we had previously is now very well organized and automated. And then once we get all the way done with the testing and the sequencing data is available, it comes back to us here in a nice summary that shows you all of the metrics from the sequencer. I won't go into them, but basically it's organized in a way that lets us very easily see if the run was successful or suboptimal. And when you highlight a sample, everything that we need to do our quality assurance checks to make sure that the data that we received is good quality data and is reasonable to report for the patient is right here in the sidebar. And we can very easily go through those tasks and efficiently sort of make sure that everything's in order. This was a huge improvement for us. One of the pitfalls, one of the benefits of next generation sequencing is you can put a whole bunch of patients together in one test. One of the disadvantages of NGS is that you can put a whole bunch of patients into one test. And so if you think about that, having so many patients in the mix here, if an error happened in the laboratory where a sample got mixed or got splashed into one of those wells that I was showing you on the plate, you could have maybe some contamination. And so we want to ensure that contamination from patient to patient sample isn't happening. And we can do that by visually looking to make sure that the same mutations aren't occurring in more than one sample. And so Sectra built out this really nice grid for us that shows pairwise comparisons of variants across the entire run. And so this was a huge benefit for us because we would do this in a very manual way previously, and now we have a very simple, very, very efficient grid that gives us that data very quickly. And then for what I do most of the time, I get the results once they're analyzed. And so this is an example of a patient's sequencing results where the mutations that were detected by sequencing are given to me in a list format. And then I have to spend some time going through collecting data on them and deciding whether they are clinically important, clinically actionable, and how I'm going to report them. And so it's not shown on here, but one of the nice features on the right-hand bar this is kind of a record, a knowledge base of the data that we've collected on a specific mutation, and we can categorize them in specific ways to convey their clinical significance. But the Sectra software actually integrates with a lot of databases that provide the information, so we can quickly sort of say, yes, this is a targetable mutation for this patient's cancer. And there's nice capabilities that allow me to filter very quickly. So I can go from a list of 100 variants to just the ones that I really need to focus on. And so this allows me to quickly curate the patient's data, annotate it, and get the results out quicker, which helps expedite any treatments the patient might be eligible for. And then finally, I won't get into the details of what this image actually is, but just to say that this is a nice feature that Fredrik and his team built out for us that allows us to visualize our genomic data in a more efficient and a much clearer way than we had before. And so enhanced visualizations tools that are just built into the software was a really huge added benefit for us in terms of interpreting complicated genomics data, which is what this plot represents. And then when we get to the reporting of the data to the EMR, this summary screen shows you on the left, there's three boxes here, 3 blue boxes that are just giving the user some visual cues that just say, hey, just make sure that you looked at everything. There's a couple things that you might want to go back and just make sure are in order so that you didn't miss anything. And they're graded, and some of these are sort of soft stops, and then we have some hard stops where it's like, no, you shouldn't sign this out because you have a variant that was not looked at that really needs to be looked at. And so this sort of user guidance that the Sectra software provides really helps minimize errors or omissions from the reports and allows us to be efficient because we don't have to go back and then amend a report or something like that. And we certainly wouldn't want to miss an important variant that would change the management of the patient. And then we have very, in my opinion, visually appealing PDFs now. And so this is kind of what the final report looks like. And then we sign it and it gets electronically transmitted across to the EMR. And this is in contrast to what we had before from FileMaker Pro. I'm not going to tear this apart because I wrote this script that generated this text-based report, but obviously the PDF is superior and it's a nice improvement over what we had in the past. And really what I want to end with is one of the huge sort of value adds that we got when we upgraded to the Sectra system and integrated with EMR. This data doesn't just go across as a PDF, a formatted PDF document. The variants go over as discrete data points that are filed into the data tables in Penn chart or EMR. And so this is what this looks like in our EMR. And so going back to EGFR, this is a lung cancer patient example where this actionable, targetable EGFR variant is very clearly displayed for the clinician. And this is a discrete data point that you can then build actually rules around in the system to provide guidance to the clinician. And so now that this data is captured in this way, we can think about making clinical decision support tools where you write rules where it says, okay, if my patient has this EGFR exon 19 deletion, I'm going to send a message to the clinician that says, hey, there are these 3 FDA-approved drug treatments for patients with lung cancer that have this type of change. And so that really is a game changer for us because it allows us to do some of these advanced feature developments that really help guide the clinician so that they're not missing opportunities to treat patients that have targetable DNA mutations in their tumors. And in addition to that, we're really now entering a phase where we can think about data analytics, not only for people who have access to the back end of PennChart, who can start integrating these genomic findings with other phenotypic and other pieces of data that exist in the EMR. And Fredrik mentioned radiology and pathology and all the other digital data that we have, but it allows the clinicians to start to understand their patient cohorts and ask clinical questions by utilizing some kind of user-friendly data analytic tools that are built into PennChart. And so this is an example where you could go and say, of all the tests that I've sent to the CPD, how many of my lung cancer patients have this specific variant? And of those, how many of them also have this additional variant? And so you can start to ask some pretty sophisticated questions. And the reason this is important for clinicians is because not only do we have the targeted therapies that are FDA approved now, but there are a lot of targetable therapies that are in clinical trials, and eligibility for those is based on, mutational profiles for the patient. And so if they can identify patients that have these mutations, they can funnel them into clinical trials so that they can, really receive cutting edge therapy, and we can move this field further. So now, with that sort of being a summary of kind of our journey and what we realized and the benefits that we're enjoying now, I just want to spend a minute to talk about future directions. I think there's a lot of opportunity for data integration, as I mentioned on the last slide, especially with digital pathology imaging. There's a lot of things that we need to visually assess about the tumor before we do clinical genomics testing on it. And there's opportunities there to automate some of that processing with integration of data across platforms. And also integrating clinical data in a more efficient way so that we can pull important milestones in the patient's cancer journey into our Sectra system. And so understanding the treatments that the patient received and the timeline in which they received them can help us contextualize their genomic profile over time, especially looking for resistance mutations to specific therapies, which we see a lot with these drugs. I'm also looking forward to building infrastructure for other diagnostic tests. I talked mainly about cancer genomics using DNA sequencing, but there's a whole other, a whole host of other areas and tests that could really benefit from the development of LIS solutions to organize this data and effectively communicate it to the clinical providers. And then, like that last slide I showed with the enhanced visualization tools, continuing to iterate off of that and find better ways to visualize our data so that we can very quickly analyze things and do more with less time is always a huge benefit. So, with that, I'm going to segue back over to Fredrik so he can sort of provide some closing notes about, how he thinks about our collaboration and our ongoing development and what we anticipate for the future.
Thanks. That was great. I think, we've learned that molecular diagnostics is important and growing, and it's also a quickly moving field that introduces new tests. It's a young field, but it's like evolving all the time. This is state-of-the-art technology, and sequencing, as Dr. Priore showed you quickly, is really cool. I urge everyone to go see that YouTube video. I find it fascinating, at least. So technology evolves, and this turns out to put a lot of pressure on vendors like us, of IT systems in this space. I think this field really requires both stable systems that's integrated with the EMR, and I think we've understood how important that is, but it also allows for efficient workflows to handle the growing volumes and needs, but yet a system that is adaptable and can change as the field evolves. Right. And for us, if we go forward, I think there's still a lot of work to be done. We've just started, of course, quite recently with this project. Although I think, I mean, it's been really, really great to work together. It's been tough for sure. I sort of smiled when you talked about the whole project, because you really evolved your own solutions with all these features, and it really took us a while to really understand how complex this is. But I think we got it together and we got a pretty good solution, but yeah, still a lot of things to do. There's a lot of things to do in every area, although I think what we've done is okay. I'm mostly interested in, of course, as you mentioned, to be able to add on more support within the field of molecular pathology, but also adding the advanced visualization parts. One thing that I think radiology takes for granted is sort of the multi-monitor support, being able to use preset rules, display protocols to direct the information to different parts of the screen and really optimize your workflow and ensure that you can sort of have a visual guide to focus on the work that you're doing, and then swap out information as you move along your diagnostic reading process. So I think that would be really cool to add. Also, if we go forth, we have now the ability to start building out these cross-discipline workflows, and I think that is huge. For one, I mean, for solid tumor testing, the upstream workflow starts at the pathologist, where he needs to identify the appropriate material to test. And I think we can build out, and we also already tried a lot with this to be able to make test annotations directly in the image in the histology slide. And finally, I mean, we already built some feedback loops between radiology and pathology. That's been hugely successful. And here I want to continue to build and add on feedback loops from the molecular pathologist to the pathologist to better understand what's a good selection to do testing, and what morphological features actually correspond to genomic genotypes. Which is, I mean, we know there's a correspondence, but I think learning to understand those and to take more decisions upstream is interesting, or be able to at least have a suspicions about them and know that this is a good case to continue with testing. So if we go forward, and next slide, please. So I think, I mean, the project doesn't end here. As you said, we're still working together. It's a moving field. From our point of view, I mean, it's been hugely successful. I said it was hard, but I'd do it again if I had the chance to do so. And I feel really honored and privileged to be able to go on this journey, and I hope, I sort of wish that more people get to have that experience, because it's been great. And if we go forward, so yeah, and next, please. So, I mean, there's a lot of labs doing this in the US, and I suspect that number is growing. Our estimate is that there are roughly 400 molecular pathology labs like yours. And we obviously want to continue to work with new customers and work with others to continue to evolve this solution. And that's really, I think, all I want to say on this. So with that, perhaps we should leave some time. I think we're a little bit overdue to open up for questions. And thank you all. And thank you, Dr. Priore.